Someone test my sound. I'm not hearing you all.
Hello.
Oh,
There we
can you
go.
hear me now?
Now I can. Yep.
Oh. It showed up the join with, it covered up the join with audio thing. How's everybody doing?
Let's see.
All right.
All right. Thank you, Connor. Thank you, Daniel. All right. Any questions from what we were going through last time? We were building the bridge to quantitative genetics so that we can understand the heritability of continuously varying characters in natural populations. Any questions?
Can you real quick just clarify what a gamete is? Is that the parent or is that the offspring?
The gamete?
Yeah.
Oh, it's neither. Gametes are sperm and eggs. So when you, yeah, I can explain this. So when you were born, this is Tessa, right? So let's assume that your father had 4 chromosomes, which he doesn't. He has 46, but just to give us more than 2 chromosomes. Your mom also had 2 chromosomes. And these chromosomes are of the same type. And so this would be chromosome 1 from your dad, chromosome 2 from your dad. This would be chromosome 1 from your mom and chromosome 2 from your dad. And we have 23 pairs of these chromosomes in our bodies, which is why our diploid number is 46 chromosomes total. We have we have 1 pair of each of 23 types of chromosomes, giving us 46 chromosomes total. We have 22 what we call autosomal chromosomes, and we have 1 pair of sex chromosomes. Now, we didn't go through the cell cycle, did we, Dr. Judd?
Not in detail. I went through it verbally, and we had a simplified diagram, and then I pointed them to the video. Okay,
so in the life of a cell is born through the process of mitosis, and then it goes through one phase of growth called G1, and then it goes through a thing called the S phase, and then it goes through G2. And during this S phase, these chromosomes duplicate themselves, okay? And so this one will create a new chromatid, this one will create a new chromatid, this will create a new chromatid, and this will create a new chromatid. And your mom's chromosomes do the same thing. They duplicate themselves so that they have enough copies of the DNA to be partitioned into new cells if you're doing mitosis, or to be partitioned into gametes if you're doing meiosis. So what happens during the first phase of meiosis is these homologous pairs. So chromosome 1, this is referred to as a homologous pair. These things line up like this, and they have the possibility of crossing over. So before this thing got duplicated, so your dad had 2 of these chromosomes. Along the line of each of these, so one of these chromosomes he got from his mom, and one of these chromosomes he got from his dad. So maybe he got, let's say, can you guys roll your tongues? How many of you can roll your tongues? Okay, so let's take the tongue rolling gene, for example. Now, I like tongue rolling because it's easy to see. So maybe your father is a tongue roller, but he's heterozygous for tongue rolling, because his dad was his mom was a tongue roller and his dad was not a tongue roller. So he ended up with this genotype, which is a heterozygous genotype for tongue rolling. He can still roll his tongue, but he doesn't have 2 dominant alleles. He only has 1 dominant allele. So when these chromosomes then duplicate themselves, it creates another exact copy, such that on this new copy, if that one was a dominant tongue rolling gene, then this one also has a dominant tongue rolling allele. And when this one duplicates, both of those are tongue rolling, except that those are the recessive tongue rolling alleles. During synapsis, these homologous chromosomes come together in prophase 1 of meiosis, which is the process of making gametes. And during that time, these things can cross over and they can undergo genetic exchange, such that now what was the end of this chromosome is now on this chromosome, and what was on the end of this chromosome is now on the end of this chromosome. So now we have those two different alleles that are now attached to different chromatids in this case. And what happens during meiosis 1 is this part of the homologous pair goes to one new cell, this part of the homologous pair goes to another new cell, and so you end up with two chromosomes that look like this. But because there are other genes located along the length of this chromosome, this recessive tongue rolling allele is now associated with a bunch of genes that it wasn't associated with before. And this dominant tongue rolling allele is now associated with a bunch of genes on this chromatid that it wasn't associated with before. Now we've taken these cells, or these chromosomes with two chromatids in them, and during the second phase of meiosis, we partition each of these into new cells. And so at the end, we've basically got 4 haploid cells, and because of this crossing over, those 4 haploid cells are all genetically different from one another, as opposed to mitosis, where the 2 cells are diploid and they're genetically identical to one another. So these gametes are then genetically different from one another. And so meiosis is the process of taking these diploid cells and creating haploid cells out of them. And each of those haploid cells only has half the chromosome. So it just has 1 copy of the 23 chromosomes. Your dad is doing this, your mom is doing that to her chromosomes also during meiosis. And when you were formed, when you were conceived, half of your mother's genome and half of your father's genome came together to create your whole genome. And so we take 1 haploid cell, we cross it with another haploid cell, and that produces now a new diploid cell that has a full complement. So when you're thinking about genetics questions, I always take it in a very systematic way. If I'm given the phenotype, I ask myself, what are the possible genotypes that those parents could have? And then once I have an idea of what those genotypes of the parents will be, the first thing that I do is I build the gametes. What kinds of gametes could that parent produce? What types of gametes could this other parent produce? So it might be easier if I just drew this on the board. So if I gave you, let's say we're dealing with peas. If I said there's a group of peas that are round and yellow. Now remember that round is a dominant phenotype for pea shape and yellow is a dominant phenotype for pea color. And I'm crossing this with a wrinkled green. And just because I've done this for a gazillion years now, I always capitalize my dominant traits and I always put my recessive phenotypes in lowercase so I can keep it straight. Well, if I tell you that I crossed these individuals and what I got was, well, now I have to work it out. I wasn't planning on this question, so just give me just a second and I'll figure out the answer. I know the answer. If this was a homozygous dominant and this was a homozygous recessive for both of these traits, what would the F1 offspring look like?
They'd be heterozygous.
They'd all be heterozygous for both traits, and so they would all be what phenotype?
They'd have the dominant phenotype, so round and yellow.
They'd all be round and yellow. But what if I told you that we got round yellow and we got round green, and we got wrinkled yellow, and we got wrinkled green, and we got 1/4 of the population was this, 1/4 of the population was that, 1/4 of the population was that, and 1/4 of the population is that. What do we now know about the genotypes of these parents? Well, the genotype of this individual here has to be what?
Both parents have to be recessive.
Both parents, so both genes have to be recessive. So we have wrinkled green, but what do we now know about this individual? Because we saw this come out as the offspring.
But they're homozygous as well, dominant.
If they were homozygous dominant, what would we get? What kinds of gametes can this individual produce?
Oh,
Somebody
just
other than Tessa. Well,
sorry.
go ahead, Tessa.
No, sorry, somebody else can go.
Yeah, somebody else do this, because I feel like I'm just having a conversation with Tessa.
They would produce all heterozygous offspring.
Yeah, this would produce this type of gamete. This individual can only produce that type of gamete. They would all be heterozygous. So when we know that this is the phenotype, all we really know, if I just give you the phenotype, is we know that there's one dominant allele for one trait, and there's the other dominant allele for the other trait. We don't necessarily know what the other copy of this homologous pair is. In order to get this group of phenotypes in the next generation, what do these alleles have to be?
Recessive?
They don't have to be recessive. Now we can test this, because we can say, well, if this is the case, from this genotype, what types of gametes do I get? How do we figure that out? Dr. Judd taught you this? I remember hearing him teach you this.
Can you just use a Punnett square? Or?
Nope.
Yeah.
How do we get the gametes? The first step is always make the gametes. Foil? What does that mean?
First, outside, inner, last.
Big R, big Y, big R, little y, little r, big Y, little r, little y. Those are the gametes that come from this parent. If this parent is the female, these are all eggs. And if this is the male parent in this case can produce what type of gamete? Only has one type of allele for each gene.
Little r, little y.
Recessive alleles for both traits. Now, Tessa, back to your question. You notice that each of these gametes has half the number of alleles that the parent had. Diploid cells versus haploid cells. We can make a Punnett square, but it's really a Punnett rectangle. We bring these genotypes, or these gamete types across. We bring these gamete types down. We have round yellow, round green, wrinkled yellow, wrinkled green. That's how you get the 1-to-1 to 1-to-1 ratio in this case. So gametes are what come from diploid organisms when they go to reproduce. So these gametes are the product of meiosis, the product of going from diploid cells to haploid cells. One of the things that students oftentimes make the mistake of doing is when they're making gametes, they don't make gametes that have one representative of each gene, one representative of each trait in it. Sometimes they'll make gametes that look like this. From this from this trait, they'll make gametes that look like this. And those aren't correct gametes. They are haploid, but what's wrong with those gamete types?
They have both alleles.
They're both alleles of what, Riley?
The same gene. So like they have the dominant and recessive trait.
Yeah, so what are they lacking actually?
Being haploid.
Well, they are haploid because there's just
Well,
two chromosomes here, but they're two of the same homologous chromosome. What are they lacking? They're lacking the other what?
The other trait.
They're not lacking the other trait. So all gametes have to have all of the traits. Another way of thinking about it is when your parents went to conceive you, when your parents produced gametes, each gamete has to have all of the traits that you possess. And the only way to do that is by each chromosome gets represented in the gamete. If one of the chromosomes gets left out and you get two copies of the same chromosome instead of one copy of each chromosome, you're lacking some of the traits, the genes for some of the traits that you possess. And so you always have to have one representative of each type of gene that you're thinking of, that you're looking at. One representative of the gene for each trait you're looking at. Does that make sense, Tessa? Does that help answer the question?
Yes, thank you.
Just so you know, I'm the king of the 5-minute explanation to a question. Other questions? Dr. Judd, do you have anything to add to this?
no, not at the moment.
Okay. Other questions? All right, then. Let me share my screen with you real quick, and we'll get this show on the road. All right, can you guys see the regression? All right, cool. So we're in the process, and now we've taken up more of Dr. Judd's time than we planned. We're in this process of bridging the gap between simple Mendelian traits and quantitative traits. So we get these quantitative distributions of traits out of simple Mendelian crosses with lots of different genes, each allele having a small effect on the overall phenotype. And so the way we measure heritability, though, is we measure heritability by just looking at the resemblance among relatives. So here we have the average offspring height and the mid-parent height, which is the average of the two parents. And we see that there is this positive correlation here. And the slope of that line is the measure of heritability. So in this equation at the top of the figure, heritability is 0.9165. And remember that heritability can go from 0 up to a theoretical maximum of 1, where the height of your parents translates directly into the height of offspring. Those are the same. The average height of the parents is the same as the average height of the offspring. Well, if we only allow tall people, tall individuals in the population to breed, what happens is, oh, and I never did change this. What happens is the average value of the parents shifts upward, and in the process of that, the expected value of the offspring generation will also shift upwards. So this represents selection, how much the parental generation is changing because of either reduced mortality in the short individuals or just an artificial breeder preventing short individuals from breeding. And that selection leads to an evolutionary response to selection. This can either be notated delta Z, or it can also be shown as R, depending on which authors you're looking at. Some people prefer using R because it stands for the response to selection, the evolutionary response to selection. Other people use delta Z because Z is usually the phenotype, and this represents the change in phenotype, delta always indicating change. But the degree to which selection gets translated into phenotypic change in the offspring is mediated by heritability. And so from this, we developed the breeder's equation, which is that selection acting on the heritability leads to an evolutionary response to selection. So if we know what the heritability is, and we know what the strength of selection was, which in this particular case is an increase of 2.41 inches, that 2.41 inches is the difference between the entire population and the population of those individuals that survived selection, or those individuals that were allowed to breed versus the entire population. So selection, S, is the post-selection mean of parents minus the pre-selection mean. So in this case, the post-selection mean is higher than the pre-selection mean, so the direction of selection is positive, and it's 2.41 inches higher. That degree of selection multiplied times this degree of heritability gives you a 2.2 inch increase in height of the offspring relative to what you would expect if selection hadn't been there to begin with. And so that's what the evolutionary response to selection is an increase of 2.201 or 2.209 inches. And so for these types of traits that have a quantitative basis, we can actually just calculate how much we can expect populations to change. This is called the breeder's equation, because imagine that you are a cow, a person who raised cows for dairy production. Turns out dairy production is determined, the amount of milk production of a cow is determined by lots of different genes, each gene producing a slightly larger increase in milk production. And if you're a farmer and you want to increase milk production in your cattle herd, in your dairy cattle herd, you could actually say, well, I want the response, I want there to be this big of a change in milk production from one generation to the next. If I know what the heritability of milk production is, I can then determine how much selection I need to exert on my cattle herd in order to get a certain response to selection. And there's a variety of ways that you can do this. You can actually, so biologists do this in a variety of ways. Sometimes biologists measure heritability and measure selection and then project what the response to selection is. Sometimes biologists will exert a certain amount of selection on a population, observe how that population responds to that selection, and then from those two things, they'll infer what heritability should be if you know selection and the response to selection, because heritability is simply the response to selection divided by the amount of selection that was being exerted. So it ends up being a very flexible way of thinking about evolution. Questions about the breeder's equation? All right. After selection has occurred, we can look at the same figure, and what we see is that these are the post-selection individuals. And one of the things that has happened as you got rid of a bunch of variation in the population is heritability went down. It was 0.9165, and now heritability is 0.6615. And so one of the things about directional selection, this type of selection, is that the population loses genetic variation over time. Dr. Judd, are you writing something on the board that we need to watch? Can't hear you.
No, I'm just getting a sense of my space for when I use it. You guys can ignore me.
Okay, all right, we're ignoring Dr. Judd for a few minutes. So they've lost genetic variability, and as a result of losing genetic variability, they've lost heritability, because heritability is just a measure of how much genetic variation there is underlying a trait. Sometimes you'll see this in the literature as additive genetic variance, and it's just a fancy pants way of saying heritability, which is just a fancy pants way of saying how is the phenotype of the offspring related to the phenotype of the parents. So in the case with the Darwin's finches, one of the things that we see in this panel, this is a panel of graphs from a selection event that occurred back in the 1970s. And what we see is the population size was really high. And the reason for this population size being really high is that this was a population size that was the result of an El Nino. So remember that when El Ninos are happening in the Galapagos, the terrestrial environment gets much more rainfall. As a result of getting much more rainfall, it produces plants, which produces seeds, and those seeds are eaten by the birds. And so population sizes can go way up. So think back about Dr. Judd's coverage of population growth models. They experience this exponential growth because there's this superabundance of seeds. But following the El Nino, things dry back out. And over time, the birds have eaten up all of the seeds, and so you see this population crash. And then we have low kind of back to background levels of the population size of Geospizophortis. During this time, seed abundance was really abundant while Geospizophortis was at high population sizes, and it declined over time to, once again, background levels of seed abundance. And this is in grams per meter squared. But what happened to the morphology of these seeds is these seeds went from being kind of intermediate sized seeds to being very large seeds to, once again, being kind of small to intermediate sized seeds. What happens is when there were a lot of birds there, they ate up all of the smaller seeds, which left behind only larger seeds. As a result of larger seeds, they were harder to crack, and only those birds who could crack those harder seeds survived, which means that over time, the beak size, the overall beak size of birds went from relatively small beaks to much larger beaks. The population after this selection event has much larger beaks than the population before selection happened. Well, what was the selective event? The selective event was this change in seed abundance and seed abundance and the types of seeds that were there. And that was ultimately driven by the loss of an El Nino event, going from a period of high food abundance to very low food abundance. And this is exactly the kind of thing that you would expect in a population that was undergoing this type of dynamic. Selection acting to increase beak size, operating on a trait that has a heritable underlying, genetic underpinning that then leads to an evolutionary response to selection, which is an increase in beak size. This is just a long-term dynamic showing you how beak size varies over time. But what I really want you to pay attention to is this. This represents, the red dots represent the variation and heritability in 1976. And you notice two things about this figure. One thing that you notice is that the range of beak sizes is quite large. What do you notice about the population in 1978?
It's a lot tighter.
Yeah, you've lost all of the small beaked birds, haven't you? You've lost this end of the population. As a result, the average parental bill length has moved up, and the average offspring bill length has moved up. And once again, this is the hallmark of directional selection. Also, this population is less variable now than the population was originally. You can exert this type of selection on, for example, oil content in corn. This is a long-running experiment at the University of Illinois. selecting for high oil content and low oil content. The reason that this selection line for continuous low oil goes down, but goes down at a slower rate than the high content oil, is that you're basically losing all of your genetic variation. So when you're down here at producing almost no oil per seed, there's really not very much genetic variation for oil production, which basically means that heritability is going down to 0. When heritability reaches 0, no amount of selection is going to have an evolutionary response to selection occurring. And this is Carnan-Penrose data. I don't think we've shown this to you before, but this is another selection surface where you have birth weight in human babies in 1951. Premature babies don't survive very well, and large babies survive less well than optimally sized babies. So this is a one-dimensional selection surface or an adaptive landscape for birth weight data in babies back during the 1950s. And what we've been doing lately is we've been through cesarean sections, removing selection at the high end, although premature babies still have quite a few problems in surviving. And so over time, we see that birth weight of offspring has been increasing evolutionarily because of severe selection at one end of the birth weight spectrum and less selection at the other end. This is stabilizing selection, but it's asymmetrical. It's stronger on the low end of birth weight than at the high end of birth weight. There are a series of other slides in this presentation that deal with snakes. Do not worry about them. We're going to revisit this topic again in the last case study, and we'll talk more about snakes later, because we're going to talk about, we can do this with one trait, but if we can do it with one trait, we can do it with 2 traits or 3 traits or 4 traits or 5 traits. And so we'll talk about a 2-trait case of doing the same type of quantitative genetic work. So cut off your studying at the Carnan-Penrose data and ignore the last 3 or 4 slides that are in this presentation. We'll revisit those in November sometime. So any questions? All right, Dr. Judd is going to talk to you about population growth.
Yep. Let's see.
I need to stop sharing.
Yeah.
All
Okay.
right, you're free to go.
All right, so our overarching theme for the next 2 lectures where I'm going to be talking to you is the idea of competition. And we're going to start with competition within species and how that influences population growth. If you've downloaded the slides already before class, I made a few updates in the last, I don't know, 2 hours or so. So if you're following along yourself or taking notes on them, want to download Moodle, got this name until the October 5th density dependent population growth. Right. So I said the overarching theme here is competition, but this is also a continuation of our discussion of population growth models. So the title for this one is Population Growth 2, a density dependent model. Let's see. Why didn't stand by? Let me redo this. Come on, share. Okay. Now we're in business. So we have a few learning objectives for this portion that are going to build on the last one. By the end, I want you guys to be able to be comfortable explaining the difference between exponential growth models versus logistic or density dependent growth models. You should be able to calculate carrying capacity. We're going to go through that today. And you should be able to explain or discuss carrying capacity in terms of competition. So there's a few vocab words. I'm going to try to give those to you guys. Make sure you know what these words mean, because you're going to see them quite a bit. So actually, you know what, I decided while Paul was talking that instead of doing this part with a slide, I'd like to do it with you all on the board behind me. So can you guys see my board okay, my whiteboard? See some nods. Okay, so we've already done exponential population growth, right? And you all know the shape of that curve. And we've talked about some of the assumptions that go with a population growing like this. As new individuals are born, the population size increases, and because those individuals have a constant birth rate and a constant death rate, at larger population sizes, more individuals are born for each unit of time. Now, initially, when we did this, we imagined that we knew the number of births between two time steps, some time 1 and some time 2. And we would fit a line to those between those 2 points. That would give us the ability to measure n of t plus 1 or t2, t plus 1 is equal to the initial population size of the previous time times 1 point whatever the rate of increase was. So we had this coefficient lambda that we talked about. It was our finite rate of increase, the rate of increase from time point 1 to time point 2. Then we wanted to make this into a continuous growth model because most organisms, like our tortoises, have overlapping generations and they can mate almost any time they want. Especially in the tropics, you'll see this. So what we did is we wanted to imagine bringing this time step 2 or t plus 1 back down the curve as close as we can to t1 so that it's instantaneous. So then instead of having this slope that intersects the curve at 2 points, we now have a new slope, a little bit shallower, usually, that intersects the curve at only 1 point. It's tangent. This value is r, little r. And that's what we call the instantaneous rate of increase. And it's instantaneous because we're taking that difference between the 2 points in the initial example, and we're reducing it down to an instant in time. And that's how you get that tangent line. So can anyone remind me, why did this graph on the left, the exponential growth model, seem unrealistic for the tortoises, such that the graph on the right seemed more realistic?
Wasn't it because like it was unrealistic because they're on an island and we didn't expect that there to be, you know, that much resources for it to keep increasing exponentially like that?
Right. We don't expect thousands or tens of thousands of tortoises after, 50 years or something like that. Less than the lifetime of an individual tortoise. So what's happening on the right is that the limitation of resources is doing one of two things or maybe both. It's decreasing the birth rate for each tortoise, the per capita birth rate, or maybe it's increasing the death rates of the tortoises. They're not all getting enough food. Birth rate maybe is a little bit more likely. These things are pretty hardy when it comes to tolerating low resources. They invest in survival in order to maximize that long-term opportunity for reproduction. So initially, we had this exponential growth model that I just drew on the board, where you can predict the rate of increase at any given time. You can predict the rate of increase of the population at any given population size, n, if you know this r value. In the logistic growth model, notice it's very similar. So we've added this new term in parentheses, k minus n over k. K in this case stands for the carrying capacity, and n is the population size at any time, any n value. So if you have k minus n, that's kind of the space for individuals, or the resources available for individuals that don't exist yet. Because if you're at carrying capacity, the population no longer increases. So you can think of this k minus n over k as the proportion of resources available that could be converted into individuals through births. Now, what range of values could this k minus n over k value have? If you plug some things in there, plug some numbers in there, what do you get? If populations are, have some number of individuals greater than 0. Or maybe another way, if carrying capacity is the maximum population size that a habitat can support, say that on the Galapagos it's 100 tortoises, and the population size of tortoises can vary between that, what kinds of values would you get for within the parentheses? So what I'm asking, can you guys see the green part here? Okay. The first one is if the carrying capacity for the island is 100 tortoises, but there's only one right now, what's the value for this term in parentheses?
$99/100.
Yeah, so near almost 1. It approaches 1. 99/100. Versus here,
100.
a really small number, right? So if we go back to the equation, this term, if this term is 1, if the population n is the same as carrying capacity, then this whole thing becomes 1, and the equations are identical, right?
So the smaller number you get in the parentheses, the more that's gonna level out, like the graph, or
Yes, right? Because if you imagine plugging in a 0 there for k minus n over k. If you plug in a 0 there, what's dn over dt going to equal?
0, right? Yeah.
Yeah, exactly. So you have no change in growth. The population isn't growing. The rate of change is 0 if the population size is at carrying capacity. So here's another way of thinking about the same thing, just using a little bit of algebra laws, right? You can just put the k under both of the k and the n, and then you get this 1, which is maybe a little bit more intuitive, minus the ratio, right? Minus the ratio of individuals to total carrying capacity. These are equivalent equations. They both do the same thing. You'll see both of them, and one of them might be more intuitive to you than the other. I want to show you one more that's going to come back to visit us in a little while. You can also create a new coefficient called the coefficient of intraspecific competition that we're going to call A in this case. It's just a variable placeholder for a coefficient here. And I've put the two subscripts 1, 1 because there's only one species in the system, and this coefficient of competition is telling us about the effect of species one, the only species here, on itself. So 1 on 1. And this value A, the coefficient of interspecific competition, takes on the value 1 over K. It's the inverse of the carrying capacity. This is going to start to matter when we have multiple species. But for now, we're sticking with just one, and I want to introduce you to this idea of the coefficient of interspecific competition is just the inverse of the carrying capacity for the population. Now, I also want to show you that when we did the exponential growth models, we were able to make that predictive by integrating it. So on the left side, you can calculate the population size at any time. If you know the starting population, you know the intrinsic rate of increase, the R value, and you know the amount of time between the start and the prediction you want, right? So this is an example of an integration that you might need to do. In this case, it's rather simple. In this case, the integration is much more complicated. We're not going to make you do this integration, but I wanted you to see that it's possible, that you can make it predictive such that if you know the carrying capacity, the starting population size, the R value, and the T value, you can predict the population size at that time T. Okay, let's think about that little r, that instantaneous rate of increase. Initially, when we were thinking about density independent population growth, what we were saying was that the birth rate here on the right and the death rate also on the right do not change as the population size increases. So if you graph it, you get a straight horizontal line. At any population size, you'll find the same death rate or the same birth rate. And R was just the difference between the birth rate and the death rate. So you get this per capita rate of increase or rate of growth rate is just the birth rate minus the death rate. And I've put this B naught and D naught, you'll see it in a moment. Basically, you can think of that as the maximum birth rate and the minimum death rate. In this case, that's the same no matter what the population size is. So we can add a density dependent component to this model by looking at the effect of population size on birth rate and death rate. So now what we're doing with these two equations is instead of just having B and D equals R, we're going to substitute in place of B naught an equation for the birth rate at a given population size and an equation for the death rate at a given population size. So the birth rate at any moment is going to be equal to that maximum birth rate minus the population size times a coefficient that refers to the strength of density dependent population growth. Now I'm using an A here as well because that's within the textbook and I don't want to deviate too far from the textbook so that if you guys are studying from that, you're seeing all different variables. So the other A I showed you that we're going to come back to, that's got the two subscripts. This one does not have the subscripts. It's just referring to this birth rate relationship. Same thing over here, except you get a different coefficient for the death rate as it relates to population size. What do you notice that's different about these two equations? Besides that one has B and one has D.
One is subtracting, the other is adding.
Yeah. Why might there be a, well, let me ask another question. Does this look familiar to you in terms of other linear equations that you've seen before? How many of you, show of hands, learned about linear equations as y equals mx plus b? Yeah, lots of those. So can anyone figure out what's the m and what's the b in this birth rate equation? Y is our birth rate, the regular b, and x is n. So what are B, and M.
Is MA
Yeah.
BY or BB?
Close. M is negative A. Yeah, negative AX plus B naught, if you want to put it in that format. Does that make sense? I see some nods.
No,
Over
can you say that
here,
again, actually?
yeah. And then over here, you can just flip it, right? Cn plus D naught. That gives you y equals mx plus b. So let's look at the actual, come on. Here we go. Wait, did I? All right. So let's just look at the left side for now. Notice what we've got, a linear equation for both the red and the blue, right? The birth rate and the death rate. One of them has a negative slope and one of them has a positive slope. So as population size increases, what happens to the death rate in red?
Increases?
Yep, yeah, you can see that because you got this rate label on the y-axis. So as population size goes up, the rate of the deaths goes up. By contrast, the rate of births goes down. That's why you've got that negative slope, negative A. Now, the steepness of the slope affects where these two lines cross. That's going to become important in a moment. Someone remind me how we calculated R using density independent growth. What was that little r value calculated from?
Was it birth minus death rate?
Yeah. Now we can do the same thing, but at any given population size, the answer is going to be different. So little r is the difference at some given population size between the blue line and the red line. So as population size increases, what happens to the height or the length of that r line?
Gets smaller.
Yeah. When these two intersect, that value of r is effectively 0. So now let's go back to the density independent model where we've got this dn dt equals Rn. If we plug in the b minus d for little r, then we've got dn by dt equals b minus d times n. Then we can substitute in the actual equations, the linear equations for b and d. Birth rate increases as population, birth rate decreases. I've got those words flipped, that's a typo, so you can correct that on your notes. Birth rate decreases as the population increases, and death rate increases as the population increases.
Nathan, it's phrased correctly because birth rate increases population growth.
Oh, sorry, I just didn't read the whole thing. Yeah, thank you.
Sure.
Yeah. Birth rate makes populations larger, right? Births make populations larger. So I've already kind of previewed this to you, but the question is, when does growth rate approach 0? Well, you can figure that out by setting this rate of change, this dn by dt, to 0, and then trying to figure out what the population size n would be at that under that condition, because of course this growth rate can vary from 0 to any positive number in the model we're looking at. So we're just setting this whole term dn by dt to 0. And if we give you the maximum birth rate and the minimum death rate and the strength of density dependence for birth rate and the strength of density dependence for death rate, those A and C coefficients, then it's a simple solve for n equation. What's the first thing? How did I get from this first equation to the second one? Anyone see how you do that? I'm showing you the steps, I'm not showing you the work.
Divided by n?
Yeah, or times 1 over n, both are equivalent. 0 times 1 over n or 0 divided by n is going to still be 0. Then you can, then you can put either then you can put the death rate equation on the other side by adding it to both sides. Then you got this where you set those two equivalent. What could you do next to isolate n?
Subtract the birth rate.
Yep, and also, oops, sorry. Yeah, you could add this an to both sides and subtract the death rate from both sides, or do it the way you suggested. It's just shuffling the parts around. So why did my animation get all weird? Okay, so here's what you end up with, right? If you, sorry, my animation messed up. I want to make sure you guys see how to make this jump here. So I'm going to make some space on the board. And I'll stop share so you can see this. All right, so you should see me again. We started here with our death rate equation on one side and our birth rate equation on the other. If you add this an term to both sides, it comes over. And if you subtract this d naught to both sides, it comes over. So then you have CA plus AN, CN plus AN equals B naught minus D naught. And then what can you do to this N to get it away?
Divide it.
Well, you can also just do the reverse of distributing it. You can just remove that N from both, and then you'll get like that. And then you get the divide step, exactly. So then you get n equals the maximum birth rate minus the minimum death rate divided by the sum of those two coefficients of density dependence. The sum of the two coefficients of density dependence. Sorry, wrong one. Desktop 2. So if it's easier to think about this with an example, we can return to Pinta Island and we can come up with a maximum birth rate and a maximum death rate for those two, for the turtles, which is the tortoises, the same ones that we used in your assignment, and plug those values into our equations. So you could end up with a graph like this, where the birth rates and the death rates for these tortoises intersect at a point that gives us a population size of 300, which we would call what? What we call that population size of 300? How is the growth of the population? What's the growth rate of the population at this intersection point?
0.
Yeah. So what do we call the size of the population that's greater than 0, where the rate of change is also 0? I mentioned it, but this is where we kind of introduce that new term. If the population is 300 and the births are perfectly balanced by the deaths, right? It's positive rates for both. The birth rates are perfectly balanced by the death rates, so the population is no longer increasing. Its growth rate is 0. We call that the carrying capacity. That's good. I saw you all. Now, if we increase the strength of density dependence on the birth rate, what number in these two equations would change if we want to increase the strength of density dependence on birth rate? We'd have to identify the coefficient of density dependence on birth rate. Which number is that? Which number is that m value negative a minus a?
0.000333. Is that?
Yeah, good, Paula. This number right here, this minus 0.000333, that's the same as the minus a in the general equation that we already looked at. So this is just the real world example with numbers in place of those general variables. So if we make this number larger, but leave that minus sign, that's going to make the slope steeper, and it'll look like this. What happened to the carrying capacity when we increase the strength of density dependence on birth rate?
Decreased.
Yep, good. This intersection point moved down or to the left along the x-axis. So the new carrying capacity is around 200. You still have a positive birth rate and a positive death rate here, but they balance each other out perfectly. So the net change, the dn by dt, is 0. Similarly, we could have left the strength of density dependence on birth rate the same and increase the density dependent effect on death rate, like this. So this blue line is back where we started, but I made the red one a bit steeper. Not as steep. I just went to 0.5 instead of 0.0005 instead of 0.0006. But still, we've moved that carrying capacity to the left. Now it's around 240. That's the new carrying capacity. So as you change the strength of density dependence on one or both of those, you can change the carrying capacity. All right, I think we'll go just another couple minutes and we'll be done. So that K value, that carrying capacity, is a measure of the strength of competition within a population or within a species of individuals that are competing. So another way to think of that is it's the degree to which a population limits its own growth as the population size increases to a certain maximum point where it limits its growth so much that its growth is actually 0. So what would happen to the carrying capacity if you reduce the strength of competition, of within species competition?
It would increase.
Yes, yep. And what would happen to the coefficient of intraspecific competition, that A11 value I introduced at the beginning, as K increases? You guys remember how we calculated that A11 value?
Was that 1/k? Is that how we did that?
Yep, exactly. Yeah, that coefficient of intraspecific competition is just 1/k. So if K is really big, what's the value of A11 going to be?
Smaller.
Yeah, really small. So that should make sense. If there's lots of resources, lots of room, then competition is going to be really weak, so that A11 value is very small. And that's saying the exact same thing as a really big carrying capacity, low competition among individuals within that population. Okay, we're going to pick up here next time, and we're going to parlay this into 2 species interacting. And I will, if there's no questions, I will see you guys on Wednesday.